Okay guys, so here we are in our next section, section 8.4, which is called RNA and Transcription. Our key concept for this is transcription converts a gene into a single-stranded RNA molecule. So big difference is we've talked about DNA. Remember, DNA is double-stranded, it has two sides, it's a double helix. Now we're going to talk about RNA, which has one strand. Okay. No, sorry, it was repeated my slide. Anyway, our vocabulary, we've got RNA, mRNA, rRNA, tRNA, ribose, transcription, RNA polymerase, promoters, introns, and exons. Our essential questions: what are the differences between DNA and RNA? You already learned one. DNA is a double-stranded thing, it's a double helix. RNA is single-stranded, one piece. So what are three types of this thing called transcription? Okay, so if we want to talk a little bit about DNA and RNA, how this whole thing works, the molecular basis of heredity. So molecular biology seeks to explain living organisms by studying them at the molecular level, the little molecules that make them up. And they use the molecules like DNA and RNA to do so. And in molecular biology, the central belief or the central dogma of molecular biology is that information is transferred from DNA to RNA to proteins. And gene expression is the way in which DNA, RNA, and proteins are involved in putting that genetic information into action, right? Because when we actually see the genes, we see what color hair you have, what color eyes you have. That's gene expression. We're putting that information that we found in DNA and RNA in those proteins into that whole organism. And basically, the genetic code is the same in all organisms. We're made out of DNA, right? That DNA gets changed into RNA, which then gets changed into proteins. Now, there are some exceptions to this dogma, the central belief, such as viruses. Viruses are really weird. They're not considered alive, even though they have a lot of features that living things do, but they transfer information in the opposite direction. They go from RNA to DNA, which is really weird. So, our belief involves three processes, okay? We have we have replication, which we talked about, DNA replication, where DNA copies itself, this thing called transcription, which we're going to talk about today, and translation. And RNA is a link between DNA and the proteins. So a little bit about RNA, so you actually know what it is. Well, RNA stands for ribonucleic acid. It's a nucleic acid made up of subunits called nucleotides, just like any other nucleic acid, right? DNA is made out of subunits called nucleotides. Now, in DNA, we have the sugar deoxyribose, right? DNA. In RNA, we have the sugar ribose. That's why it's ribonucleic acid. Now, RNA can be thought of as a disposable copy of a segment of DNA. And we'll talk about why a little bit later. The RNA base sequence, so the order of those bases, directs the production of proteins. And ultimately, those proteins result in our phenotypic traits. Phenotype are just physical traits, so what you look like. Now, if we compare RNA and DNA, how they're alike? Well, they're both nucleic acids. They're both made out of long chains of nucleotides. We've got some differences. The sugar in RNA is ribose, and the sugar in DNA is deoxyribose. RNA is single-stranded, not double-stranded like DNA. And RNA doesn't contain the same bases that DNA does. It contains almost all the same bases, but instead of thymine, instead of the T, it contains a U for uracil, okay? But it contains everything else, just not the T. So in RNA, if we have an A, it goes with U, not T. Okay, so A and U go together, but we still have a C and a G. So RNA synthesis, or how is RNA actually made? Well, making RNA or RNA synthesis takes place during a process called transcription. And in transcription, little segments or little pieces of DNA are copied to produce complementary RNA molecules. Remember, complementary from when we talked about DNA replication just means that they match. Now, where in the cells does this take place? Well, prokaryotes, that happens in the cytoplasm, right? They don't have a nucleus. In eukaryotes, RNA is produced in the cell's nucleus, and then it moves into the cytoplasm. Now, transcription makes three types of RNA, and here they are. The first type is this thing called messenger RNA. We abbreviate it using a little m, so a little m RNA. And it does just that. It carries a message that's going to be translated to form a protein. Then we have ribosomal RNA, which we abbreviate with a little r RNA. And this actually is what makes the ribosome. It forms part of the ribosomes where those proteins are actually made. And then our last type is transfer RNA, which we abbreviate with a little t RNA, and it brings amino acids from the cytoplasm to the ribosome to actually help to build that protein. Remember, proteins were built out of amino acids. Now we do have 3 stages of transcription. In our first stage, the enzyme RNA polymerase binds to DNA, and it basically does what it does in DNA replication. It separates the DNA strands. But now we have RNA polymerase, not DNA polymerase. So once it separates, then we have RNA binding to these places called promoters, or regions of DNA that have specific base sequences. One is going to say start and one's going to say stop, and we'll learn about that a little bit later. Okay, and RNA is edited before it's used. So portions that are cut out and discarded are called introns because it doesn't use the whole thing, it doesn't need it. So it gets double checked, it looks at it, it edits it, it cuts some pieces out, we call those pieces introns. The remaining pieces get stuck back together, okay, or spliced back together, we call those exons, and those form the final mRNA message. Now the RNA strand then detaches from the DNA once that gene gets transcribed, because this is the process of transcription, and then it gets sent out to the cytoplasm where it will find the ribosome. And then the ribosome is actually where the proteins get built.